Determining the absolute configuration of an alcohol. This experiment will be a little different than the types of activities you have performed so far in this course. Instead of learning a new technique such as spectroscopy or thin layer chromatography, you will now be applying techniques you already know to answer some questions. In this experiment, you will answer 2 questions in regards to an unknown molecule that you will be given. By the end of this experiment, you will determine the structure of the unknown molecule via proton NMR spectroscopy. You will also determine the absolute configuration of that unknown molecule. You will do so using what is called the CEC method, which applies TLC. You will talk more about the CEC method shortly. To start today's webcast, let us talk a little bit about chirality. If you are concurrently taking Chem 232, you have recently covered this topic. For a brief review, chirality has to do with the 3-dimensional structure of a molecule. Here on this slide, we have 2 molecules that are identical, except for the 3-dimensional orientation of some of its components. As you can see, as drawn, these structures are mirror images of each other. If you try to take the structure on the right and overlap it with the one on the left, you will find that you cannot be successful. These mirror images are what we call non-superimposable. This means the stereochemical relationship of these 2 molecules is that of enantiomers. Enantiomers are non-superimposable mirror images. So, we have a pair of enantiomers here. Why do we care? We care because the chirality, or more specifically the stereochemistry of a molecule, can have tremendous impact on the biological applications of that molecule. For example, this molecule on the left is the S enantiomer of what we call naproxen. You might know naproxen better by its trade name of Aleve. Aleve is an analgesic that is used as an anti-pain, anti-fever, and an anti-inflammation pharmaceutical. Now compare those biological properties to the biological properties of the molecule on the right. Our naproxen is a liver toxin. You can see how knowing exactly which enantiomer of a compound we have is important. For this experiment, you should review your Chem 232 notes on a few topics. You should know what stereocenters are and how to identify them. You should know the rules to assign whether a stereocenter is R or S. And you should know how to determine if a molecule is chiral if it contains stereocenters. Remember, a molecule can have stereocenters but not be chiral. Mesomolecules are an example of this. Given the importance of distinguishing one enantiomer of a molecule from another, just how easy is it to distinguish the enantiomer of one molecule from the other? Unfortunately, enantiomers have the same physical properties. For example, 2 enantiomers of a single molecule would give us the same proton NMR spectroscopic data. They would have the same retention factor on a TLC, they would also melt at the same temperature, boil at the same temperature, and have the same solubility. They do, however, rotate plane polarized light in different directions. Unfortunately, that does not help us determine which enantiomer of a molecule we have. The direction a compound rotates plane polarized light does not tell us which enantiomer it is. It only tells us that we have a chiral molecule. Fortunately, there are techniques that allow us to identify the absolute configuration of a molecule. X-ray crystallography is a very common technique used by chemists. In your biology courses, you may have seen crystal structures of various proteins. Those crystal structures were generated using X-ray crystallography. The motion method is a technique organic chemists often use to establish the absolute configurations of molecules that contain alcohols. Vibrational circular dichroism is a more recent technique that utilizes computational methods to try and predict the absolute configuration of a molecule. All of these techniques require specialty equipment and a significant amount of time and training. The technique you will be using is what is called the competing Enantioselective conversion, or CEC for short, method. The CEC method, as you will see, is quite simple to execute and to analyze. To discuss the CEC method, we will break the CEC method into its constituent parts. We will talk about the competing portion, the enantioselective portion, and the conversion portion separately. To start, we shall discuss the competing aspect of this method. In this experiment, you'll be taking an alcohol, shown here on the left, and turning it into an ester, shown here on the right. This figure here is what is called a reaction scheme. This may be the first time you have seen something like this. A reaction scheme is how organic chemists communicate what is being used in a chemical reaction. On the left side of a reaction scheme, we have our starting materials. These are the chemicals, the reagents we are going to use in our chemical reaction. We have this arrow moving from left to right that designates that our starting materials on the left are being converted into what we call products on the right. Everything on the right side of this arrow is a product. Located above and below the arrows are where organic chemists often place what are called the reaction conditions. The reaction conditions can include things such as other reagents being used in the chemical reaction. In this case, we are including a type of molecule called an anhydride, in this case butyric anhydride, and down below, an organic base, in this case triethylamine. Other information you might find above or below an arrow in a reaction scheme include things such as the reaction temperature, the reaction time, and the reaction solvent. Let's interpret this reaction scheme here before we continue. What this reaction scheme tells us is that we are taking an alcohol and using this anhydride and this base, we are converting our alcohol into this ester. This type of reaction is called an esterification. If you are concurrently taking Chem 232, you will not have covered this material yet. That's okay. For now, you simply need to be able to read a reaction scheme where starting materials are on the left, products are on the right, and above and below the arrows are the reaction conditions, and be able to identify the type of molecules being used. We have an alcohol being turned into an ester using an anhydride and a base. This type of reaction often proceeds very quickly. However, under the particular conditions you will be using, this reaction is very slow. To accelerate or speed up this reaction, you will be using a catalyst, shown here in blue. This particular catalyst is called homobenzotetrabenzol, or HBTM for short. Do not worry about memorizing the full name, just recognize this molecule as being called HBTM. Now that we know what kind of reaction you will be performing in this experiment, let us discuss how you will be using it. In this experiment, you will be performing this esterification reaction twice. You will perform two separate reactions. Each reaction does the same thing. Both reactions take an alcohol, and using an anhydride and a base, convert that alcohol into an ester. This reaction is accelerated through the use of a catalyst, HBTM. If you look closely, you might see that there is a slight difference between these two reactions. If you look at the HBTM catalyst, the reaction up top uses one enantiomer of HBTM, and the reaction on the bottom uses the other enantiomer of HBTM. This is a subtle but very important difference. To recap this slide, the competing portion of a CEC experiment comes from you performing two identical experiments, except one experiment uses one enantiomer of HBTM, while the other experiment uses the other enantiomer of HBTM. Before continuing, let us discuss how HBTM accelerates this reaction. Here on this slide, I have HBTM, and I am exposing it to the anhydride used in this reaction. What's happening here? When HBTM is placed in the presence of an anhydride, it reacts with that anhydride to make what is called a reactive intermediate, which you see here on the right. The anhydride on its own is not reactive enough to react with the alcohol. What we have done is taking two low-energy reagents and combine them to make one high-energy reactive intermediate, which will react very quickly with alcohols. We have finished discussing the competing portion of a CEC experiment. Now let us discuss the Enantioselective portion. Here on the screen in blue is the structure of HBTM, our catalyst. However, you know that this structure is not complete. You recognize that there is a stereocenter here at this carbon connecting the nitrogen and the phenyl group. This means this molecule is chiral and comes as two enantiomers, shown here in black. One of these is the R enantiomer of HBTM, and one of these is the S enantiomer of HBTM. I'm not going to tell you which one is which. I expect you to be able to assign which enantiomer of HBTM is R and which one is S. Okay, so HBTM is chiral. Why do we care? We care because while enantiomers have the same physical properties, they may and often do have different chemical properties. In this experiment, you will take each enantiomer of HBTM and react it with one enantiomer of alcohol. In this case, let us assume that your unknown alcohol possesses this stereochemistry. Here on this slide, we have a chiral alcohol that will react with a chiral catalyst. Will this chiral alcohol react the same way with each chiral catalyst? The answer is no. We will see what we call Enantioselectivity. The chiral alcohol will be selective for one enantiomer of catalyst over the other. Let's talk about why that is. On this slide, I am showing a pair of gloves. At the top, I have a left-handed glove, and at the bottom, I have a right-handed glove. Think of these gloves as the HBTM catalyst, where the left-handed glove is one enantiomer of the catalyst, and the right-handed glove is the other enantiomer of the catalyst. Now, let us take these gloves and try to fit a hand in them. In this case, my left hand. You can think of my left hand as the one enantiomer of alcohol on the previous slide. We're going to take one enantiomer of my hand and try to place it into each enantiomer of this glove. Or, in the actual reaction, we're going to take one enantiomer of alcohol and react it with each enantiomer of catalyst. There are two potential outcomes. The first outcome, taking my left hand and putting it into my left-handed glove, results in my left hand fitting in my left-handed glove quite well. In the other situation, taking my left hand and trying to fit it into my right-handed glove does not work out so well. I can get it in there, but it doesn't really fit right. This top situation is a matched case. The enantiomer of my hand matches the enantiomer of the glove. The bottom is what we might call a mismatched case, where the enantiomer of my hand does not match the enantiomer of my glove. We can take this same analogy and apply it to our two chemical reactions. Here we are showing our activated intermediate already formed. Consider this top activated intermediate as one enantiomer of glove, and this bottom activated intermediate as the other enantiomer of glove. Each activated intermediate will react with the same chiral alcohol. Now remember, we don't know which enantiomer this alcohol is. That's what you were trying to figure out. But you do know you have only one enantiomer. This alcohol is the same as my hand on the previous slide. What will happen here is the alcohol will react with the activated intermediates to form the product ester. Both reactions form the same product to regenerate the catalyst, and a triethylamine salt that was used to deprotonate or remove this proton that was on the alcohol. One of these cases will be what we call a matched case. The other case will be a mismatched case. What determines whether the top reaction is the matched case or the bottom reaction is the matched case? The source of the enantioselectivity of this reaction has two components. The first has to do with the stereochemistry of the catalyst. Here on this slide, I am showing two visual representations of our alcohol reacting with our activated intermediate. On the left, I have a two-dimensional drawing of this reaction, and on the right, I have a three-dimensional drawing generated using a program called Spartan. In both images, my alcohol is at the top, and the activated intermediate is at the bottom. In this drawing on the left, I have drawn this phenyl ring here, pointing down, in a red color. If you look at the three-dimensional structure here on the right, you see that this phenyl ring points straight down. The consequence of this is the phenyl ring effectively blocks the bottom half of our activated intermediate. If our alcohol wants to react with the activated intermediate, it has to come from the top side, because the bottom side is blocked by this very large, very bulky phenyl group. That is the first component of this reaction's enantioselectivity. The second component of this reaction's enantioselectivity has to do with the aromatic system on the catalyst. If you look to the drawing on the left and look at the blue-colored portions of the activated intermediate, we see an aromatic system. This aromatic system participates in what is called pi stacking with the aromatic system on the alcohol. In your biology courses, you may have heard about pi stacking in relation to DNA. What is happening here is the aromatic portions of the molecules are lining up to stack on top of each other. You can think of the catalyst as a kind of landing pad for the aromatic ring on the alcohol. What these two components do is, in the matched case of the enantiomer of alcohol and the enantiomer of catalyst, the alcohol is perfectly positioned to react with the carbonyl. In a mismatched case, using the other enantiomer of our activated intermediate, this OH would not be perfectly positioned and would take more time to move and rotate in order to react with the carbonyl in our activated intermediate. I want to make a distinction here. The phenyl ring on the stereocenter is not involved in the pi stacking with the alcohol. The phenyl ring on the stereocenter is entirely a steric interaction. It's big, so it blocks one side of the activated intermediate. It is not involved in the pi stacking that orients the alcohol for its attack on the activated intermediate. That finishes our discussion on the enantioselective portion of this method. Now let us discuss the final conversion portion. Okay, so we have two reactions. Each reaction takes the same alcohol to make the same product. The only difference between these two reactions is the enantiomer of catalyst used. One of these reactions will be a matched case where the enantiomer of the alcohol is matched with the enantiomer of the catalyst, whereas the other reaction, the enantiomer of the alcohol, will be mismatched with the enantiomer of the catalyst. To explain why this is useful to us, we have to go back to general chemistry for a bit and talk about kinetics. Recall that kinetics deals with the rate, the speed of a reaction, and that the rate of a reaction depends on K, which is a constant which varies from reaction to reaction, and the concentration of the reagents. For this particular reaction, the rate law is K, our constant, times the concentration of the alcohol, times the concentration of the catalyst, times the concentration of the anhydride. If these reactions are identical, shouldn't they have the same concentration of alcohol in catalyst and anhydride? The answer is yes. Both of these reactions are using the same concentrations of these reagents. Therefore, shouldn't these reactions proceed at the same rate? The answer is actually no, and this is because of enantioselectivity. The matched case will be faster. Here, in our competing reactions, one reaction will be faster than the other because the faster reaction will be the matched pair of alcohol and catalyst. And it turns out, if we know which reaction is faster, we can identify the absolute configuration of our alcohol. If the reaction using our HBTM is faster, then the alcohol, when drawn like this, has the OH coming out towards us, which here is the S configuration. If the S enantiomer of catalyst is faster, then our alcohol is the R configuration. Okay, so we're going to have a matched reaction and a mismatched reaction. The matched reaction will be faster. If we know which reaction is faster, we know what enantiomer of alcohol we have. How will we determine which reaction is faster? To do this, we will look at conversion. Conversion is a measure of how far our reaction has proceeded, how much product has been made. Have we made 25% of our product, or have we made 75% of our product? In this experiment, both of your reactions will run for the same amount of time, 1 hour. This means that the faster reaction will produce more product. Let's talk briefly about the procedure. To recap, you have 2 reactions. Both reactions use the same unknown alcohol. Mechanically, this experiment is quite simple. You will take pre-made solutions and add them in the correct volumes in the correct order. The correct order is very important. Once you have added all of your solutions in the right order, you will then have to wait 1 hour. In that 1 hour waiting time, you will take proton NMR spectroscopic data given to you and determine the structure of your unknown compound. Your alcohol will be one of these 3 options, shown here on this slide. You will then in your notebook justify which structure is your unknown compound. Now remember, enantiomers give the same spectroscopic information. Therefore, you will not be able to determine which enantiomer you have using the NMR data. That is why you are performing the CEC method. In this experiment, you will determine which reaction made more product using TLC, which we will discuss on the next slide. Using this data, you will then establish the absolute configuration of your unknown alcohol and justify your conclusion in your notebook. Here is an example CEC Analysis TLC plate. We have 2 lanes, A and B. Each lane is 1 reaction. We see that in lane A we have 2 spots, and in lane B we have the same 2 spots. However, the spots in lane A have different intensities than the spots in lane B. How are you going to interpret this TLC? Remember, in your reaction you are taking an alcohol and converting it into an ester. One of these spots is the alcohol and one of these spots is the ester. You will need to determine which spot is the alcohol and which spot is the ester using what you already know about TLC. Once you have identified which spot is the ester, you will then look and see which lane has the more intense ester spot. The intensity of a spot is directly proportional to the amount of that compound present. More intense spots, darker spots mean more material. You will look at your 2 lanes and see which lane has the darker ester spot. Alternatively, you could also look at which lane has the lighter alcohol spot. Because remember, the alcohol is being used to make the ester. The faster reaction will use up more alcohol. The alcohol spot should be lighter as a result. You will notice that this plate is green. In order to visualize these spots, you'll be using a new method of TLC visualization. You'll be staining your plate using a stain called phospholipid acid, or PMA for short. Simply follow the instructions in your procedure. The post-lab quiz for this experiment will be a little bit different than the post-lab quizzes you have seen so far. Thus far in the course, you have learned new techniques, and the post-lab quizzes tested your understanding of those techniques. This experiment is teaching you about a new technique, the CEC method, but it is also applying old techniques. For this post-lab quiz, you should expect some fundamental questions on stereochemistry. Be able to answer questions involving the stereochemistry concepts I specified at the very beginning of this webcast. You should expect some fundamental questions on the CEC method. You should understand the competing aspect of the CEC method, the Enantioselective aspect of the CEC method, and the conversion aspect of the CEC method. You should also expect critical thinking questions. What do I mean by critical thinking questions? This experiment is about taking techniques you know and applying them to answer a question. What if I changed something about the experiment? Would you be able to predict what would happen as a result? That's what I mean by critical thinking questions. Taking what you know and being able to predict what would happen if something was changed, or being able to decide on what to do if you wanted to cause a particular change to occur. Finally, expect spectroscopy questions as well. This experiment is not about spectroscopy, but it does utilize spectroscopy. And given the importance of spectroscopy, you should expect some questions testing your knowledge of it. That brings us to the end of this webcast. To recap, the CEC method allows us to determine the absolute configuration of an unknown alcohol. It does so by comparing the rates of two reactions between the alcohol and a pair of catalyst enantiomers in the presence of butyric anhydride. Which of these two reactions is faster is established using TLC. After knowing which reaction is faster, we use a predictive mnemonic to establish the absolute configuration of our unknown alcohol.